Okay, so I'm happy to be the host today for the NannoTalks Volume 2. This is a new monthly schedule we are going to have, and I would like first to thank Aura Sounds Communication and especially Felipe for all the organization he has made. He will be in charge of the technical aspects today. If you have any issue, just chat to him directly. The NannoTalks Volume 2 is organized with Aurora by let's say a committee that is composed of Felipe Valero, I'm not sure I'm pronouncing that well, Clara Bolton, Sarah Alvarez, Alicia Khan, and myself. We are planning monthly talks, and we want to focus on recent interesting publication that can be interesting for, let's say, wide aspects in nanoplankton research. And also we'll be inviting young scientists to be able to talk about their research and to meet the community. Today, we let me check before I didn't forget anything. Yeah, and of course, I would like to thank Juliana Villa that just arrived. I saw that. I would like to thank her to support this NannoTalks Volume 2. And we'll be communicating through her for the next NannoTalks. We have not planned the date yet, neither the speakers, but we've done all the scheduling in the following, I would say, days, week. I think I'm fine. I didn't forget anything. Clara, maybe any comments? No, that's fine.
No, I think you've said everything.
All right, thanks. So we can now go to the presentation today. We have invited Gerald Langer because he has published a recently in New Phytologist, if I remember well, this study called Role of Silicon in the Development of Complex Crystal Shapes in Coccolithophores. We have then invited him, and just before he will start his presentation, I will just remember a few aspects of Gerald's career. So Gerald Langer is specialist in coccolith calcification biology. He has been working quite intensively on living in cultures of coccolithophores, and he has been recently quite interested also in holococcoliths. He's been working for the Marine Biological Association in Plymouth for the past 5 years as a research associate, and has been working through time in the UK, Spain, and Germany. And then I will give him I will let him speak about this presentation he's going to do. It will last for 15 to 20 minutes. Just if you have any questions, you can write your questions in the chat room, whether on YouTube or in Zoom. The Aurora team will send me the question, and at the end of the talk, I will manage the Q&A. So I will read the questions that will be in the chat, or if someone wants to take the speak, they must ask me before, please, in order not to have everybody speaking at the same time. I think we are also done on that part. So Gerald, are you here?
Yep.
So I leave you now the talk.
All right. Thank you very much. And I'll try to get my talk on the screen. So I hope that works. Does it work?
It does indeed.
Okay, great. Thanks again, Baptiste, and thanks to the NannoTalks organizing committee. It's a very good idea to do these talks. And yeah, as Baptiste said, I will speak today about the role of silicon in coccolithophore calcification. Now that might sound strange to some of you because, as you all know, coccoliths contain calcium carbonate, precisely calcite, and usually silicon is associated with diatom pustules, and not many people have thought about silicon in coccolithophores and coccoliths. And this is because until recently, it was unknown that coccolithophores might require silicon to calcify. That was actually first published in 2016. It's a study by Christina Durac et al. And in this paper, they showed that some, not all, some coccolithophores need silicon to grow and calcify. And they could clearly show that they need silicon to calcify, but they left the question open why they need silicon to calcify, because calcification in coccolithophores, as you might know, is a very complex set of processes, and there are many, and they range from calcium transport, cellular calcium transport, cellular inorganic carbon transport, to vesicle transport, exocytosis, and a lot of other things. The morphogenesis of coccoliths is in particular is very complicated, and we know relatively little about it. You might remember Jeremy's talk, and he spoke about that. So we have all these processes, and it was quite unclear where silicon comes in and what its role might be. So that's what this talk is partly about. I should say partly, because I give you a warning, for the first half of the talk, I'll not talk about silicon at all, which is strange, but I hope that will become clear as I go along, because I want to make a few other points which will be helpful in understanding the role of silicon. And that was something which came together quite nicely when we try to find out more about silicon. So enough of the introduction. I go right into the thing we're interested in here, which is coccolith crystals. And coccoliths are very complex structures. They're very beautiful. And if you listen to Jeremy's talk, you know all about them. And this is quite a lot. So we know a lot about their structure. And they have these beautiful crystals. You can see here on the right, left-hand side, actually, as an example of Coccolithis parodiae. And on the right-hand side, you see a couple of other example images, coccoliths and the coccospheres. So we have these crystals. And what's interesting about them is they have various shapes and sizes. So that's quite amazing, because calcium carbonate, to shape it in that way is difficult. So that's the question is really, how is that done? And I say these elements you can see here, for instance, the V or R units, you can see on the left-hand side, upper left corner, are crystals, are single crystals. Now that's debatable. And I will not go into detail now, but if you want to find out more about this very important question, it's not so important in the context of this talk, but it is important if we are to understand coccolith formation and coccolith crystallization. So this is a review paper came out recently, actually a couple of days ago, and it states the question and states the importance of the questions and possible ways to solve it, I think, for the first time. And so check it out if you want to know more about whether coccolith crystals are really single crystals. But for the moment, I call them single crystals. So we're talking about these crystals, but actually, not all of them have these really intricately shaped crystals, these big crystals of various sizes and shapes, there is another life cycle phase. As you probably know, I'm not going to go into any detail here, but you probably all know that the so-called heterococcolith life cycle phase produces these heterococcoliths with the big crystals, different shapes and sizes. You can see here an example of calcidiscus, famous one. So the heterococcoliths in brown, and but below them you see these smaller coccoliths, the holococcoliths, and they are composed of these tiny crystals. And this is quite different from what the heterococcoliths produce. And this is amazing because it's the same species. It's just a different life cycle phase. So if we look at the holococcolithophore crystals in a bit more detail, we see that they are small, they are all of the same size, and all of the same shape. They are rhombic, which is interesting because it looks comparatively simple if you compare it to the heterococcolith crystals. And also, it is a shape which is typical for inorganically precipitated calcite, you can see on the lower right. So this is a shape which you do not need to do much to get it. You can precipitate it in a beaker, and the example you see on the lower right is a lab precipitated calcite, basically in a beaker. So that's easy in terms of how you shape a crystal, do the morphogenesis. And so that's very, very different from the heterococcolithophores. And that seems to fit because the generally accepted idea is that the calcification process of heterococcolithophores and holococcolithophores are totally different. So whereas the heterococcoliths are produced intracellularly in specialized vesicle, highly controlled chemical environment, and so on, the holococcoliths are supposed to be produced extracellularly on the surface of the cell in a less controlled environment and in a totally different way. And that would fit the simple crystals which you can produce in the beaker, so why can you not produce them on the surface of the cell? And there's also the idea that because these mechanisms are so totally different, and because we find in the fossil record, we find the holococcoliths later than the, so they appear later, so they're younger than the heterococcoliths. That means that there must have been a reinvention of calcification by coccolithophores. So they reinvented it totally new from scratch and a totally different mechanism. So that all seems to make sense if you look at these simple crystals. However, and now I come to the first bit of our study, which was looking at holococcolithophores and how they calcify. So contrary to what I just said, holococcolithophores actually do calcify intracellular in a vesicle in very much the same way as heterococcolithophores do. And you can see here transmission electron microscope images showing this. So the top two images show 3 crystals on a scale in a vesicle, in a cell. In the middle, you have various crystals. So well, I should say that the top image shows holes in the resin. That's why they're white. They're not crystals. There are holes in the resin. In the middle image, however, this is black. These are real crystals. They're not fallen out of the resin. So I'm not going into technical details here. If you don't know about TEM preparation and so on, don't worry about that. Just take my word for it that these are crystals inside the cell. And you can also see these yellow things in the middle right picture, which show you a so-called energy dispersive X-ray spectroscopy analysis, which is a way where you can use that in order to say something about the elements in your structure. And so it has a very strong calcium signal. So it's pretty clear there must be calcium carbonate. So these are calcite crystals, and so we have two different species here. The top images show coccolithos, and the lower one shows calyptosphera. Right, so this is quite a change in terms of what we thought we knew about holococcolithophore calcification. It completely changes the picture because it's not a totally different mechanism, fundamentally different, but actually it's very much the same mechanism. So the fundamental things that it's inside this vesicle and then exocytosed upon completion is very much the same. So we have to rethink that bit of the story. And I just mention that the discovery that heterococcoliths, I should say, are produced inside the cell and then upon completion extruded. It goes back to Dixon in 1900. It's a top right sketch you can see. That's what he did at the time using his light microscope. And you might remember that from Jeremy's talk. So I just wanted to mention that because it gives you an idea. So if you consider that coccolithophores were first described, I think it was in the late 1860s. So after some 30 years, Dixon discovered that heterococcoliths are produced inside the cell, but actually it took another 120 years to find out that holococcoliths are also produced inside the cell. So that tells you a little bit about how difficult it is to find that. And it took us a long time. Now, there's another line of evidence that these holococcoliths are produced inside the cell, and this is the chemistry of the calcite. Just mentioned that that if you produce calcite in seawater, it has a relatively high magnesium content, and in the top picture you can see an EDS analysis of the magnesium of an extracellular calcifier, marine calcifier, the foraminifera amphystigina lysonii, and it has a clearly detectable magnesium peak. However, the coccoliths of both coccolithos and calcidiscus, they don't have any detectable magnesium peak, and neither the heterococcoliths nor the holococcoliths. So that again shows you that it's highly unlikely that this calcite was produced in contact with seawater. So I come now to silicon. If we want to study the need for silicon in calcification, we have one powerful tool, which is germanium. Germanium is an analog, it's a silicon analog, and it kind of blocks the silicon, is a competitor, and it kind of blocks silicon uptake. So we can use it to simulate silicon starvation, because silicon starvation is actually very, very difficult to achieve, and I come back to that a bit later. But what you can see here is growth rates measured under germanium influence and of various holococcolithophores, and they don't show any difference to the control. There's no effect. They don't need silicon. I show the example of Syraxphara pulchra because it's a bit different than the others. It's very sensitive to germanium. And looking at the panels in B, the left-hand side, you might get the impression, oh, if you use germanium, then you have this lower growth rate. So that's kind of that's something. Well, I should start actually with a panel in C, because the holococcolithophores of Syraxphara, they are much more sensitive to germanium than the other holococcolithophores, calcidiscus, coccolithos, calyptosphera, and so on. So that might give you the impression that the holococcolithophore is sensitive to germanium, i.e. needs silicon, but the comparison you can see here clearly shows you that that's not the case. So the germanium silicon ratio of 1 affects the growth rate a bit, but they still calcify completely normally, which you can see in the pictures down there at the bottom. And this is a ratio which kills all other, all heterococcolithophores, indeed. And if you compare that to the hetero Xeroxphara pulchra, you can see a clear difference. So if you grow them, the heterococcolithophore in a germanium to silicon ratio of roughly 0.1, it has a massively reduced growth rate. In the holococcolithophore, that's not the case. And calcification is unaffected, so that's very important. So the take-home message here is holococcolithophores don't need silicon. And remember, they have these simple rhombic crystals. So if we look at heterococcolithophores by contrast, they do, some of them. And I not go into too much detail about which do and which don't. So for the present talk, I would like to focus on the ones which do. So here's an example, Coccolithophora hoodii, and if grown under germanium treatment, which mimics silicon starvation, you can see that the percentage of malformation, coccolith malformations increases. And that's interesting. That, in principle, has been documented before in the Dirac paper in 2016. However, we analyzed the coccolith morphology in a bit more detail, and we found that actually some of the malformations you can see under the germanium treatment are specific. They only occur if you treat them with germanium. Other stressors will not produce these kinds of malformations. And we call them ROM-like and ROM malformations. The ROM malformations is in particular interesting because it's basically a calcite ROM. And you might think, yeah, well, how do you know that's actually supposed to be a coccolith? We do know for several reasons. I not go into all of them. I just give you one of the reasons, which is it has a typically low magnesium content. So if it would be an inorganic precipitation from the seawater they're grown in, you would expect the high magnesium, remember? So this is one of their other reasons, but we are absolutely certain that these are coccoliths, and they look like an inorganically precipitated calcite. Now, you can see with this example that there's something wrong here with crystal shape. So these coccoliths, they have malformed crystals. So that's very striking. I give you just one other example, which is Xeroxphara, Xeroxphara pulchra, sorry. So here we have an effect as well of the germanium treatment, and it also produces several types of malformations. Among them, the germanium-specific malformations you can see in the top. This is a malformation which is very, very rare in other samples. You would probably never find it. So that's why we call it specific. So if you treat it with germanium, you find it in these percentages, which is incredibly high compared to what you find in other samples. And now, if you look at the plot in B, that shows you one example of a low silicon seawater experiment. As I said, these are very difficult to do because to get that seawater, natural seawater, I should say, this low silicon concentration is very, very difficult. And the 0.2 micromolar we used here is just about what you need. If you go a little bit higher, they will be perfectly happy. But here we can see that in the 0.2 micromolar silicon seawater, silicate seawater, I should say, you get these specific germanium-induced malformations, which we call the type C. And this clearly shows that the effect of germanium is not some kind of generic effect which, you know, poisons whatever, and then you get these malformations, and it really got nothing to do with specific calcification processes and so on. So I think, and silicon and all the rest of it, so there might be this argument, but I think the low silicon experiment shows that that's not really the case. So the take-home message here is that it's the complex crystal shapes of the heterococcolithophores which require the silicon in some way to be formed. So the coccolithophore cell needs the silicon in some way, not at all sure how and how exactly, but that's the point where it comes in. So it's nothing to do with say, calcium transport or something like this. They can produce massive calcite structures. That's not the problem. So we really have a specific morphogenetic effect here. And it is these complex crystal shapes and the simple rhombic crystal shapes of the holococcolithophores. They don't need this process requiring silicon, including silicon, whatever the exact process is. So to finish, I just want to give you a slightly revised idea of how coccoliths could have evolved. This is again, I shouldn't say against. This is an alternative version to what you probably have heard before, which is that the heterococcolithophores, the heterococcoliths came first, and then the holococcolithophores were later reinvention, as I mentioned. And what we now would like to suggest is that probably there was a non-calcified ancestor which produced these organic scales intracellularly, and they performed some of the functions in terms of protection that later coccoliths would. And then what happened was at some point these scales, or whatever you call them, were calcified with simple rhombic crystals like we find now in the modern holococcolithophores. And that basically was the stage where holococcolithophores developed step by step. And then later there came in a mechanism which allowed shaping these little rhombic crystals into big, variously shaped crystals, and allowing to form such structures as interlocking coccospheres, coccospheres of the, let's say, placculus bearing species such as Calcidiscus. And this silicon-dependent mechanism allowed this, and this is where the heterococcoliths and heterococcolithophores come in. So they utilize this silicon-dependent mechanism. And some lineages lost it later because not all of them need it, as I mentioned, but I don't say no more about that now. So I leave you with that, and thanks for listening.
Thank you very much, Gerald. What's brilliant presentation. Thank you very much for this presentation. If there is anybody that wants to ask a question, you can add it in the chat room, or if you want to ask the question yourself, just tell me, hands up, and I give you the right to ask your question. Anybody wants to? Yes, Jeremy?
Yeah.
Oh, sorry, Jeremy, and then Ines.
Nah, lovely talk, Gerald. Where's Gerald gone? He's not. I guess you haven't got your video, get your camera on. But no, that was really nice, fascinating stuff. So, what do you have any feeling for how the silicon how the silicon works in crystal shaping? Do you think can it act as a cytoskeletal inhibitor in some way, or do you think it's changing something like the polysaccharides?
Yeah, that's a very good question. Thank you, Jeremy. I try to get myself on the video. Give me a second. I hope that works.
Yay! Hi, Gerald.
Okay. Hello. Great. Yeah. Thanks. That's a great question, and I think we can be fairly sure that it's nothing to do with the cytoskeleton, because comparing. So we did another set of experiments where we used cytoskeleton inhibitors on coccoliths and Skiffsphaera absteinii, and compared the malformations you get to the malformations you get from germanium, and they're totally different. So this suggests that it's really nothing to do with the cytoskeleton, and I would indeed say your suggestion with the polysaccharides is a very good one. That's actually my favorite idea. So I think that must be it. And the way I see it, which is pure speculation, is that the silicon is a structural component of one of the polysaccharides used in morphogenesis of the coccoliths. And so that's not completely wild idea, because it's known that silicon is a structural component of some macroalgae polysaccharides, so that in principle such things exist, and I think that's a way to to look a bit more into in the future.
Thank you. Then Ines was. Thanks.
Yes, hello everybody. I have one question regarding silicon. I didn't understand quietly. Does silicon give malformation to coccoliths, or you said something on the not last night, the picture before that, we can see some kind of malformation. But I'm not sure what did you want to say? That slight malformation of, is it when silicon in the marines water column are increased, then it's much higher malformations? I didn't understand quietly.
No, actually, it's the opposite. So if you starve them of silicate, I should say, in the water. So in the water, you normally, as you all know, you have a certain silicate concentration, and there's a huge range. And in natural seawater, if it's very, very low, it goes down to values round about 0.2 micromolar. And that's actually, if we use that in an experiment, if we get seawater that low in silicate, then we find these interesting malformations, which you also get if you treat them in an experiment with germanium, which is this analog inhibitor thing. So this is a kind of poison which specifically acts to block silicate uptake, and so the idea here would be they need silicate, they need silicon, they take up silicate, but they need silicon in some way to produce normal coccoliths. So if you take that away, then they will not produce normal coccoliths, and there will be these malformations, and they will in some cases be very specific to the silicon starvation. So does that answer your question?
Yes, because I was studied lots of marginal marine environments, and I didn't found out malformations of coccoliths in assemblage with diatom, silicoflagellates, and other silicon species that developed their parts from silica. And I totally agree with you. I thought I understand differently that you said in your presentation. Thank you.
All right.
Thank you, Ines. Juliana was wanted to ask a question, so you go.
Yes, okay. Can you see me?
Yep.
First of all, I want to thank you Gerald, for this innovative discussion, very interesting. And also, I want to thank for the organizer, Baptiste, Philippe, Aurora, Alicia, all the guys that organized this new session of NannoTalks. That is very important to keep alive our community. And of course, you, Gerald, for to be the first of this run of talks. My very quick question is do you think can be an influence of, sorry for the dog, an influence of volcanism and so emission of silica in some part of the ocean for the nanoplankton? I remember in some paper, I think of Bakri, the paper that he was mentioning that this Quaster Sinius was more abundant in the area of volcanic activity. And that could be something related to silica emission?
It's a very difficult question. So I think what we not entirely sure, well, actually what we know next to nothing about is what is the does silicon starvation in the oceans out there in the field really cause a problem? Because as I said, in order to starve them of silicon, just by lowering the silicate concentration, you have to go very low. And you have to go so low, in fact, that it's just about what you get in natural seawater in some places, sometimes. So there's this example, which you can see in one of, I think it's in Louisa Cross, is like she has this guide to the Western Med, this beautiful book essentially with all the cockles. You probably know it. And there actually is a picture of Syracusphira pulchra coccolith, which has the germanium specific malformation. And so it comes from an area and a time of year in midsummer where the silicate concentration can be as low as 0.2 micromolar, which is exactly what we used in the experiment to produce the same type of malformation. So I think in those areas and at those times, you getting close. In all other areas and at all other times, it's probably not really an issue. That's my gut feeling. We know very little, and as I said, it's very hard to judge. So all I can tell you is that the danger comes if you go to the area of 0.2 micromolar. If you're higher than that, they are perfectly happy.
Thank you. Thank you again.
Thank you, Juliana. So George wanted to ask a question. Do you want me to read your question, George? Yeah, that's right, I guess. Okay, no answer. So I will read it myself. He asked what made you start your study with silica and not other elements, and he's asking if there is the same behavior with any other elements you might think, or I don't know, you suppose that might be the effect of other elements the same way as silica.
It's a very good question. And I'm not sure I can satisfactorily answer it, but I would say this. So the first question is easy to answer. So what got me started on silicon? It was basically the Durac 2016 paper and the chance I had at the time to go to the MBA in Plymouth, where they done this study. So that was basically it. I was talking to Colin Brownlee about it, and I was immediately hooked. And I said, "Yeah, that sounds super interesting. Let's do it." So that's the answer to that. The second question is more difficult, and I think that we can't really single out another element like that at the moment. there's this very special case where we had the idea that it might have a kind of morphogenetic function, which is strontium, in fact, in Sciphosphira absteinii. So I should say that strontium in Sciphosphira absteinii is unusually high. It's much higher than in other coccolithophores. And so the idea was maybe it's got a morphogenetic function there, but it hasn't. Apparently it hasn't. We did an experiment and lowered the strontium concentration, and there was no problem. In fact, if you increase the strontium concentration in the seawater a lot, then it becomes a problem. So it doesn't seem to be in any way involved in the morphogenetic process in the same way silicon is. So I would say I can't give you, at the moment, I can't give you another element like that with such a modulating function. There will be a lot of elements involved in general, but that's more general. I mean, you can point to the general cell physiology of magnesium and so on, but that's not really the same thing, I guess. That's very different. So yeah, that would be my answer to that.
Thank you very much, Gerard. Patricia wanted to ask you a question.
Oh, and thanks a lot for the presentation. Sorry I was a bit late, but I didn't want to miss it. So actually, it's very great to see you and also the other that I didn't see for a while.
Hi, Patricia.
So I wanted to ask you, I mean, malformation is a very complex topic for coccolithophores, and I guess we started to know more about malformation when all these experiments on ocean acidification were basically developed, and then we saw the high CO2. In some cases, we had malformation. So the first question is about the type of malformation that you detected when you do an experiment, acidification experiment. Is it comparable or is different than the one that you see in low silicon conditions? And the other one is like, how much you know about this species-specific response to this limitation in terms of silicon limitation?
Well, the second, I'm not sure I get the second question completely, but I come back to that in a second. The first question is an interesting one, and as far as we can say now, so one big problem I have here in answering that question is that, as you know, as you well know, probably better than many other people, that most of the ocean acidification work was done on EHACs, and we have a problem that EHACs does not need silicon. So that you can't compare that. So that's a problem. So what do you compare? And the best candidate would be coccolithos. So, and luckily I can compare that because we have an as yet unpublished data set we're working on at the moment. It was done by Doro Kottmeyer at the MBA, and she looked at effects of acidification in coccolithos in more detail, and we also analyzed the morphology. And I can tell you that this is a different story. So the malformations you get from germanium are very different from the ones you get from acidification. So this is a different mechanism. I'm pretty sure I can say that. Okay, I hope that sort of answers your question. It's very hard because we don't have a lot of data, but watch this space. I hope we will be able to submit the acidification manuscript soon. The second question, so I think, do I get this right? The second question is, what do we actually know about how silicon works? Is that right?
Basically, yes. Practically, it's like how much we know about the species-specific need of silicon.
Oh, all right. Okay. Species-specific, yes. So what we know is that there seem to be species which don't need silicon, and they're not completely random. They kind of phylogenetically they cluster. So EHACs and G5 capsa, they don't need silicon. Pleurochrysis carteri doesn't need silicon, and you have a few of those. and they come in these clusters. So that's why we get the idea that it was probably originally a mechanism which all heterococcolithophores used, and then it was lost at several times in evolutionary history. So that's the idea. So it's species specific, yes, and there's a bit more species specificity, I think, to that. In terms of what kinds of malformations you get, but that's a long story, and we'll hopefully publish a separate paper about that. We have much more data, so this is something we want to look at in more detail in the future. But yeah, there is specific, and so the general idea would be you have that as an original requirement, the silicon, and then it gets lost several times.
Yeah, can I ask something else quickly?
Yeah, go, and then it will be Jeremy. You finish your question, and then Jeremy.
Sorry. So you mentioned the work of Louisa in Western Med and possible malformation there. I was wondering, like, the challenge of really looking at natural environment and malformation there, and then be able to detect different type of malformation depending on the condition. Because first of all, I guess they are very rare, because you mentioned the conditions are very, very unusual for the natural environment. So I guess you think you are confident enough that you can detect different malformation, and then say what type of malformation, like in terms of silicon.
I think, yeah, I mean, yes, in some cases. So certainly the ones I presented. So in coccolithos, especially in Syracusphira, it's very easy. And there's another example, Scypsphyra absteinii, is also very, very easy to detect that. So if you find in a natural sample, if you find these malformations, they're easily recognizable, and you can directly relate them to silicon starvation. So that's very easy. It's not the case in all silicon requiring species, I should say, but that's, as I said, these are different data which will hopefully come out later. But yeah, so there are differences. But we have at least 3 species which produce these very specific malformations you would recognize in an environmental sample, yes.
Okay, thank you. So Jeremy wanted to ask you another question. You go.
Yeah, well, it just seems an extraordinary coincidence. And do you think it is that Pleurochrysis carteri and Gephira capsomeliania don't need silica, and they're also the ones which don't produce phyllococcoliths? The other ones are unrelated. It's an amazing coincidence, isn't it?
It is. We thought about it, but well, I can't tell you much more about it actually. But it is interesting. I'm not sure exactly what it means, but yeah, that's right. So in these species, the typical species where you have this silicon dependency, they need to calcify in the hetero phase. And also they calcify, they don't necessarily need to, but they do calcify usually in the hollow phase. So that's right. Yeah, that's all we can say. You're right. I mean, it is interesting.
Have you tried testing in Hymenemonas then? Yeah, one more test of whether that correlation holds.
I think we actually didn't, as far as I remember.
Might be worth just pushing it to see if that correlation really holds as you go through that.
If we did, let me think. We did a few other species, and I think I'm not sure at the moment. Sorry.
Yeah.
Okay. There is a question from YouTube, so I will read it for you. It's from Barney Balch. Is anything known how the holococcolithophores regulate the shape of individual holococcoliths in stacking the rhombic crystals into very regular specific structures? Thank you.
Thanks. That's a great question. I'd love to know the answer. but I can't tell you. We don't know. But it is one of the, I think it's one of the big questions. And I remember discussing that question with many people, especially Jeremy, over the years. And yeah, we simply don't know, unless Jeremy has another idea. But it is very, very tricky. And I think that how I imagine is that in principle, the morphogenetic machinery of holococcolithophores and heterococcolithophores is probably very similar. So you get this cytoskeleton, the idea that the cytoskeleton exerts a force on the vesicle membrane and so on. I think that holds for holococcolithophores as well, and that must be part of it. But yeah, it's I can't tell you. It's one of the big questions, I think.
Oh, sorry. Okay, thank you. Amos Winter wanted to ask you a question. Amos, your microphone is off.
Can you hear me? Can you hear me?
Yeah.
Hi, how's everybody? Nice to see you all. Just a question, maybe a stupid question, but I just looked at the history of silica, and you know, it was like 80 milligrams per liter just at the start of the Jurassic, and has gone down to nearly 0 today. I was wondering if you think that has any implications for the evolution of holococcolithophore evolution. Yeah, because right now we're lowest it's ever been.
Yeah, I think that.
How does that fit in?
That's a good question, and I'm not sure really. I mean, there are two things here. So one is, as I said, not all heterococcolithophores, they do need silicon, so it was lost later. So I think maybe that's to do with the lowering of the silicate concentration you mentioned, but also it's low, but again, if you look at silicate concentrations in the present day surface water, it's not that low. So in most places, at most times of the year, these coccolithophores, the heterococcolithophores requiring silicon, should still be okay. So I'm not sure how great an influence that has. It relates to this ecological question. So what does it mean for ecology and blooms and so on? So I think I'm not sure, but you're right. I mean, the fact that it goes lower is certainly can certainly become a problem. Whether it has been a problem over the last, I don't know, 100,000 millions years is very hard to say. So I'm not sure.
When there was a switch from, what did you say? It went from hollow, I can't think anymore, hollow to hetero. No, from hetero to hollow, and then there was a switch. When was that?
Well, I don't know. I think what happened. I mean, what we just suggesting is that the hollow evolved first. So you go right back to the beginning of coccolithophores, and the first things you have are hollow phases, and then later the heterococcolithophores come in. I think that can be timed because we have the first appearance of heterococcolithophores quite early.
Anyone know here in the audience what happened?
Quite early. We go back to I think certainly more than 100 million years, right? So that's quite early, and but the point is, I'm not sure because we have this drop in silicon at some point, in silicate, I should say, but it's not at all clear whether that had any kind of effect on the evolution or the calcification of the species at the time. So I think that's a tricky one. And my gut feeling is the silicate concentrations were over geological time way too high to make a difference in terms of what we know about present day species in terms of their requirement. That said, it could be that in the past, the requirement was higher in the sense that they needed higher silicate concentrations, and that might have, but that's all speculation. And since we don't know, I can't really.
I understand, but just something to think about.
Yeah, absolutely. It is an interesting point, and it would be good to look into this bit more detail, but it is tricky. So yeah.
All right. Thank you very much.
Sorry.
Thank you, Amos. There is another question from, I'm sorry for the name because I don't know how to pronounce it, but Eric Chang, sorry for the name. But anyway, it says, great talk. I was wondering, did you check whether the morphology of the base plate was affected upon germanium treatment?
We didn't, but that's something we are looking into right now. So yeah, hopefully can tell you in the future. But at the moment, it's not clear. I think the first impression we get from a few preliminary data is that it might be affected, but that's not in any way statistical, and I can't really confirm that. So basically unknown.
Okay, thank you very much. I guess we are out of question, and it's been an hour now, so I think we can close it for today, I guess, except if there is a last question from anybody.
One last bit.
Okay, Jeremy.
So this 0.2 micromoles, is that about the level which diatoms reduce silica to, if they, because fundamentally silica levels are low in the oceans when they've been stripped out by diatoms, and diatoms will reduce silica down to a level where they then can no longer extract silica. So is that 0.2 micron level essentially the level which diatoms reduce silica to?
That's right.
And so then which might suggest indeed that coccolithophores have had to evolve to that level.
Yeah, exactly. I think that's right. They certainly, in fact, not all diatom species can do that. So if you use different species, some of them, they will not go down to 0.2 micromolar, but some of them will. So it really depends, but certainly they cannot go any lower. And it's hard for them to go to the 0.2, whereas with the coccos we looked at, you have to be at least as low as 0.2, preferably lower, but it's very, very difficult to get that. So yeah, so I think you're right. I mean, there is a difference. Coccolithophores are much more efficient. They can take up silicate much better than diatoms can.
Especially in the North Atlantic, there's a sort of general ecological story of basically the diatoms flourish during the spring bloom until they reduce silica level down to the level where they can no longer, and then coccolithophores are able to kind of, they're still nitrate and phosphate, but the silica's going down. So,
yeah. Yeah.
Okay. I think there is no more questions, so I would like to conclude. Oh, wait. There is just a last question that came out. Let's say it's the last one for today. I guess it's coming from YouTube. Could the silicate concentration in Mesozoic ocean be related with the emergence of muralites? And the decrease in silicate could be related with the placolitic emergence? It's a tricky question, I would say.
That's a very tricky question. I would say no. For one reason, basically, which is that the silicon the amount of silicon in coccoliths is very, very low. We haven't published the numbers yet, but we will hopefully in the near future be able to do that. And but I can tell you it's very low, and I think it's just not sufficient. But I haven't done any budget calculations, I should say. So that is basically a gut feeling based on a very, very low amount of silicon in coccoliths. But yeah, so.
Thank you. So I guess we can stop here for today. The discussion was great, indeed. It opens a lot of direction, but the study was great and the talk was great. That's why everything went fine, I guess, at the end. A great discussion. And I'm quite happy, actually, we have this great discussion because the NannoTalks Volume 2 was indeed, it was done to make the community get back together because we are not been able to see each other for a long time. INA will be postponed from 1 year, so we will not be able to see each other in real life. I don't know how to say that until next year. So that's great. We can have this discussion, and your study was really interesting and make us connected and discuss in different people having different specialties. I would like to thank everybody, and especially Gerald, that was the first one to do that this year. We will send the next speaker will be chosen in the following days. We'll have the next talk in May. We will send you the time and the date as soon as possible. I thank again the committee that is selecting the talks. So Clara, Sarah, Alicia, and Felipe. I would like to thank especially Aurora Science Communication through Felipe, Alejandro, and Daniel that were planning and managing all the technical stuff today. And I would like to thank Juliana Villa, the president of INA, that allowed us to do this NannoTalks Volume 2. And I guess I'll see you, all of you, next month for the second talk of NannoTalks Volume 2. Thank you, Gerald. Thank you, everybody, and see you next month.
That was very good.